The structure of contractile proteins, primarily actin and myosin, is complex and crucial for muscle contraction. Let's explore this in detail. 1. Actin thin filaments. Structure. Actin filaments are composed of two helical strands of filamentous actin F-actin twisted together. Each F-actin strand is a polymer of numerous globular actin monomers G-actin. These filaments also incorporate two regulatory proteins, tropomyosin and troponin. Tropomyosin is a long, fibrous protein that winds along the groove of the F-actin helix, while troponin is a complex of three globular proteins troponin T, I, and C that are positioned at regular intervals along the tropomyosin. In a relaxed muscle, tropomyosin, regulated by troponin, blocks the myosin binding sites on actin. 2. Myosin thick filaments. Structure. Myosin filaments are composed of numerous myosin molecules bundled together. Each myosin molecule is a dimer composed of two heavy chains and several light chains. Each heavy chain consists of a globular head heavy meromyosin, hem, and a tail region light meromyosin, LMM. The hem head contains an actin binding site and an ATP binding site, along with an ATPase enzyme that hydrolyzes ATP to provide energy for muscle contraction. The hem heads project outwards from the thick filament, forming cross bridges that interact with actin during contraction. These cross bridges are arranged in a staggered pattern along the filament, allowing interaction with actin along the entire length of the overlap zone. The LMM tail regions form the core of the thick filament. Conclusion. The intricate structure of actin and myosin filaments, along with the regulatory proteins tropomyosin and troponin, is essential for the precise and efficient functioning of muscle contraction. The interaction between these proteins, driven by ATP hydrolysis, generates the force needed for movement and other bodily functions.